Hello everyone, my name is Isaac Cherian. So I think we can just start the section for today. So everyone else will join afterwards, and I will just present the slide for them. Okay, so good evening. So today we will start the week 4 section for the course Cell Biology, Cell Organization, Division and Process. So we will discuss about 30 questions for today, 10 questions from previous NPTEL session for this course, and previous year NPTEL session for this course, and 10 questions from the current NPTEL course, and 10 questions that are currently made from the video lectures that I have seen for this that has been uploaded by the NPTEL. So I think we can just start the first question for today. Sorry, 1 minute. Okay. So coming to the first question, which protein complex holds sister chromatids together until they are separated during mitosis? Cohesin, condensin, separase or kinetochores? You can either type in the chat box, or you can unmute yourself or give the answer. Okay, I'm getting option number A. Any other option? Okay, so you are basically right. It's a basic answer, right? The answer to this question is cohesin. Cohesin is a protein complex that hold together sister chromatids until they are separated during mitosis, right? So basically, if you look into a second option, condensing, condensing is a protein that helps the DNA to condense together to form what is called as a chromatids, right? So that's not the basic answer there. And also separase is an enzyme that actually clears out this cohesin. Basically, separase is actually a protein that is bound to securin. So securin is actually an inhibitor for separase. So before the anaphase promoting complex is active, from the cyclin B is active, this is inactive in nature because the separase is bound by this separin, which is acting as an inhibitor. When the APC is active or anaphase promoting complex is active, the securin is cleared out and separase become free, and this separase acts on the cohesin that is bound to the sister chromatids, so and cleaving it out. So when the cohesin is cleared out, the complex sister chromatic gets separated out, and then it transition from the metaphase to the anaphase stage, right? Because the chromatids are free, and then it can move to the poles, and then cytokinesis can happen. So kinetochores are actually structures that are bound to the centromeres, right? So it helps in attachment of microtubular assembly, so that the spindle formation can happen. Once spindle formation can happen, the metaphase stage initiated, then it is moved into anaphase, where the sister chromatids are moved onto different poles, and afterwards telophase happens. And once telophase is completed, cytokinesis happen, and two new cells are formed from one parent cell by a process called as mitosis, right? So this whole cycle is called as mitosis. So condensing is the protein complex that holds sister chromatids together until they are separated during mitosis. So if you look into the structure of cohesin, this is the structure of cohesin. You can see that it is a clip-like structure held together by this particular protein called as SCC1. So the clip-like structure consists of this dimeric structure called as dimeric protein structure called as SMC3 as well as SMC1. So SMC stands for structural maintenance of chromosomes. So basically this two clipper-like structure actually binds to the chromosome at the centromere, and then it is locked in place by this SCC3 group protein. So what context is done is that if you look into a budding yeast and vertebrate, it is almost similar in both cases. So there is the cohesin that is binding together the two chromatids in place. So when this CDC5 in case of budding yeast is active, or when this particular kinase or mitotic kinases which helps in the transition from metaphase to anaphase is getting activated, what happens is that the cohesin gets phosphorylated in this case. So when the coercion gets phosphorylated, it becomes loose, and what happens is that this separase then, which is activated by anaphase promoting complex, upon release from securing molecule, cleaves out the remaining, that is the phosphorylated coercion or the loose coercion in this case, so that the chromatids get separated in this case. So that promotes the conversion from the metaphase stage to the anaphase stage. So you can see that the securing, which is inhibited by inhibiting the separase, is getting inactivated there. The separase cleaves out this coercing molecule, and then the two cytochromatic get separated. And the two processes are more or less similar in this case in budding yeast and cellulose vertebrates. So they are basically kinases, right? So it phosphorylates certain kind of dissolve this inhibitor molecule, thereby helps in the metaphase to anaphase stage there, and thereby cleaving out this SEC1 molecule from this RAT21 particular protein molecule that binds, interlocks these two dimeric proteins together from the coercin molecule, thereby relieving the two sister chromatids. So I hope this answer is clear. So anyone has any doubt? Okay, so I think it's clear. So I will move on to the next question then. Okay, coming on to the next question. Centrosomes are composed of two perpendicular structures known as chromatids, spindle fiber, centrioles, or microfilaments. Which may be the answer? Centrosomes are composed of two perpendicular structures known as Okay, so the answer to this question is centrioles. So basically, this is the structure of the centrosome here. This is the whole centrosome complex there. You can see that this one is a centriole, this one is also a centriole, and this entire space that is occupied by these centrioles is okay. So anyone has any doubt? Okay. So it is occupied by a space called as pericentriolar matrix, right? PCM, and there are interconnecting fibers that connects the two centrioles together, and to each centriole microtubule attachment is there, and it helps in microtubular nucleation. Basically, in this case, formation of microtubules take place from centrioles that are present within the centrosome. So this PCM consists of several proteins that helps in this microtubule nucleation and assembly also. So basically centrosomes are composed of two perpendicular structures known as centrioles, you can see here, and it is found in animal cells, acts as a primary microtubule organizing center, or MTOC, and are involved in cell division. Not involved in directly involved in cell division, but directly involved in microtubular nucleation and assembly, and indirectly because of that cell division happens in that case. So you can see the two centrioles here. One will be considered as a mother centriole, and the other will be a daughter centriole, because the centrioles have a tendency to divide. The centrioles have a tendency to divide from each case. So one will be considered as a mother cell from where the mother centriole from where the daughter centriole is getting formed, and each centriole is connected by interconnecting fibers there. The centrioles are also surrounded by a pericentriolar matrix here. You can see the matrix here, which consists of proteins that helps in microtubular nucleation and assembly, and also involved in nucleation and anchoring of cytoplasmic microtubule that we have discussed this time. And each centriole is consists of a 9 triplets of microtubule arranged in a parallel structure. So if you look into this particular section of a microtubule, you can see that it consists of this particular cylindrical structure, more or less a cylindrical structure. So if you look into this particular component, this is called as a microtubule, and this microtubule consists of a triplet. So there is a triplet of microtubule there that forms about this particular structure, and these are about 9 in number. So if you count it there, there are 1, 2, 3 microtubule that forms a microtubule triplet there. And how many microtubule triplets are there? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 microtubule triplets are there. And there is a central hub there, and there is also a spoke there. The spoke protein actually attaches the central hub to the microtubule tribule, and each microtubule triplet is attached to each other by a AC linker there. So basically, if you look into this microtubule triplets, each microtubule triplet is called as a protofilament there. So this entire microtubule triplet is called as a protofilament there. So this is the structural aspect of a centriole. So it consists of a 9 plus 0 organization, meaning that there are 9 protofilaments that are comprising of the centriole, and there are 2 centrioles there forming a 2 into 9 plus 0 arrangement of this particular centrosome there. So basically centrosome, and these are the perpendicular structure. These act as a perpendicular in nature, and hence they are called as centrioles. And it is a component where tubulin components are present there. From the tubulin components, because of the charge nature of the tubulin components, microtubule assembly take place in this case, and helps in and hence that's why it helps in microtubule nucleation and all. So centrosomes are composed of 2 perpendicular structures known as centrioles. So anyone has any doubt? Okay, so I think it's clear, right? So I will just move on to the next question then. So coming on to the next question. Which motor protein are involved in the movement of chromosome during mitosis? Dynein and kinesin, myosin and actin, tubulin and actin, and keratin and myosin. Which may be the answer? There are only 2 motor proteins that are present there, right? Subtypes are there, but as far as we know, only 2 motor proteins are there at this stage, right? Which may be the Okay, I'm getting option number 3. Sorry, but the answer to this question is option number 1, dynein and kinesin. So basically, these are motor proteins that can help in movement of cargo proteins. That if you look into a structure, you can see that. So this one that you can see at the end are called as dynein, and at the center position you can see that these are kinesin, right? So these motor proteins can actually move along the microtubule. These are microtubule region. You can see that there are centrosomes there. So it is negatively charged, and hence the centrosomes, the negative charge of the microtubule actually point towards the centrosome. So the opposite end of the microtubule, that is positive charge consisting of this GTP or ATP molecule, helps in polymerization and all. They are formed towards the poles because of this charge disparity there. So this will be negative end of the microtubule will be focused towards the centrosome. In this case also you can see that it radiates out. The positive end of the microtubule actually radiate out from the centrosome. So if you look in this dynein, that is which is shown in green, actually occupies the the poles, more probably majority at the poles, while the actin occupies the kinesin actually occupies the center. And these are basically motor proteins because they have the tendency to slide or move along the microtubular assembly there. So it can move from one end to the other. So basically, if you look into a dynein, it is actually negatively charged. So dynein has a tendency to move from the positive charge to a negative charge, because majority of the dynein molecules are minus n directed or negative n directed. Kinesin, majority of them are plus n directed, that it is moved to the region of the microtubule where polymerization is taking place because of the presence of GTP or ATP at the plus end. However, there are exceptional cases where there are also kinesin that can move towards the negative end also. So in this case, these are motor proteins, right? So we will discuss about that later. So these are basically motor proteins. So what happens is that because of the tendency to move or slide, because of the tendency of them to move or slide along the microtubule, they can carry cargo from one position to other. Suppose that there is a protein that formed from this particular region here, and it needs to move to this region. So what does these motor proteins do? It carries this cargo from this end to the other end. That is one function. And also in case of cell division, whenever the chromatids need to be separated out, suppose that this microtubule attached to a chromosomes there, these dynein-kinesin as well as dynein molecules cooperate with each other. They function with each other so that the chromatids can move from the equator of the cell to the poles. That's how metaphase to anaphase transition take place, right? So these motor proteins also have a tendency to carry out this process, because these motor proteins can change the orientation or change the movement of this microtubular assembly, so that the chromatids can separate from one end of the that is the equator to the pole, so that the cell division or cytokinesis can take place afterwards. So that's the basic function of the this motor protein. So basically, if you look into kinesin, kinesin consists of several other proteins. That's why it's called as a kinesin superfamily, that is KIF. Consists of over 40 proteins divided into 16 families. More than that are there. And they participate in creating bipolar mitotic spindles. Bipolar means that there are positive and negative charge at each end, right? So they can help that formation of these particular mitotic spindles there. Promote chromosomal congregation to spindle equator, and interacts with chromosome to generate a force that move chromosomes away from the poles. So these are positively charged, so they can move to the positive end. Because of this particular friction or the force that is created there, the chromosomes have a tendency to manage at the equator there. And also plant cells also, and several other KF include KF11, 15, 1, 22 and 4, that are essential for spindle organization and chromosome movement during prometaphase and metaphase. And that you know, right? Basically, because of this motor protein, the chromosome getting segregated there. And also plant cells also utilize kinesin-like motor protein to build cell plate and new cell wall for during mitosis. So in plants, there is a particular region called a cell plate that is formed before cytokinesis there, right? So cell plate helps in the formation of the furrow there, and then cell divide accordingly because of the formation of this cell plate there. In case of animals, there is something called as a contractile ring there, right, which leads to a formation of a cytoplasmic furrow or a cytoplasmic cleavage there, which helps in cytokinesis. So these are two different processes that are taking place in animals and plants. However, they lead to one common effect, that is cell division itself, that is formation of two different cells from a single progenitor cell. While in case of kinesin, if you look into they are also motor proteins that helps in intracellular transport towards the minus end of the microtubule. Why? Because they are minus end directed, they only move to the minus end of the microtubules there. And they are composed of heavy as well as multiple heavy and light chains, like what you see in myosin, right? Myosin and actin, they are heavy as well as light chains that are present there. And also in case of antibodies, some heavy and light chains are present there. But these are two different functionalities. These proteins carry different functionalities, right? But we can compare based on the names given there. So it also, the cytoplasmic dyning contributes to a poleward movement of chromosome during mitosis. Chromosomes need to move to the pole, right, because it is negative directed, minus end directed. This exert a force so that the microtubules move to the pole regions along with the chromosomes where it is attached to a kinetochore, right. So that's the basic functionality of this motor protein. So I hope it is understood. So anyone has any doubt? Okay, so I think it's quite clear, right? So we'll just move on to the next question then. So coming on to the next question, the contractile ring that facilitates cytokinesis in animal cell is composed of microtubules, intermediate filament, actin and myosin, tubulin and myosin. Which might be the answer? We have discussed that before, right? It's actually found in animals, not in plants. So what might be composed of? It need to contract, right? Yeah, I'm getting option number 3. Any other option? Okay, so basically you're right. The option number 3 is the answer. Actin myosin. So it is also called as an actomyosin contractile ring because it is composed of the two ring. Which if you combine to it, then it becomes actomyosin. So basically this is the whole process there. So what happens is that rho GTP, the rho is a protein that can actually bind to both GTP as well as GDP. It can hydrolyze the GTP to form a GDP. So basically if the rho is to be activated, then it should be bound to GTP itself. So several proteins like MGC and ACT1 helps in this process. So MGC actually is a guanine exchange nucleotide factor that helps in the transfer of this GTP to GDP in this case. So the rho is inactive in this stage. But ACT1 is actually a protein or nucleotide exchange factor that helps in the conversion of this or the replacement of this GTP to a GDP to a GTP. Thereby the rho is now bound to GTP, so it is active there. So this interrelation that actually helps in the what you say activation of the or the leads to a formation of initiation of contractile ring there. So in first cases when it's inactive, you can see that there isn't much active state there. You can see that the microtubules are bound to the chromosomes there. You can see that it is somehow like positive polar charge because of the presence of the microtubules there, right? Because much of the end will be negative charge in this case. When the rho protein is bound to GTP, it is active in nature, right? So what happens is that it gets localized into this particular region there. So this particular region is called as rho active zone or rho activity zone in this case, and this signals that there is a need that the contractile ring needs to be formed there, and you can see that the chromatids are also separated from this region, right? So this is the anaphase stage because you can see the chromosome chromatids getting separated to the poles there, and the rho GTP is getting accumulated there, and it is active in nature. And afterwards, not only that, when it is active, it initiates the process of activating actin as well as myosin filaments there. So what it does is that in one case it activates this formin and it binds to this particular profilin actin ATP. This profilin helps in polymerization. So when it is bound to ATP as well as recognized by this particular protein component called as formin, it binds to the ends of actin that is already present in the binds to end of actin, which is microtubule that is present there, and then it leads to actin polymerization. And also other than that, rho GTP activate this particular kinase there. So this particular kinase phosphorylates this particular protein and also this particular protein, myosin phosphatase. So myosin phosphatase, if it is active, it inactivates this particular protein. So once it is phosphorylated by this kinase, rho kinase or citron protein, what happens is that it becomes inactive in nature, and it also phosphorylates this RMLC to a phosphorylated form, so it is active, and this in turn activates the myosin molecule there. And then these two once both of them are active, actin as well as myosin are active, they form what is called as an actomyosin there. You can see that here in this case this is the myosin, this is the actin there. It combines together to form what is called as a contractile ring. You can see that there is a contractile ring that is formed because of the activation of this rho GTP there, and other components are also present there. Don't need to go into detail there. So afterwards, when it is formed, it starts to constrict because of the actin disassembly that is taking place. The actin is getting shorter there, so it contracts accordingly. So when it is contract, it leads to a formation of what is called as a cleavage furrow there, and this is what's seen in case of animal cells. They are not in plant cells. Plant cell, it leads to a formation of cell plate, not contractile ring. So it leads to a formation of cleavage furrow there, which helps in promoting cytokinesis. You can see that it enters into a kind of a telophase stage. The chromosomes are getting like decondensed there. You can see the mass there, and hence it leads to cell division. So this is how the cytokinesis take place by this contractile ring formation there. This is the whole step that is taking place in between. So the contractile ring that facilitates the cytokinesis in animal cell is primarily composed of actomyosin or actin and myosin. So I hope this answer is clear. Does anyone have any doubt? Okay, so I think it's quite clear, right? I will just move on to the next question then. Okay, so in plant cells, cytokinesis is accomplished by the formation of which structure? Cell plate, cleavage furrow, mitotic spindle, or centriole? Which may be the answer? Option A. We have discussed this right now, right? Yeah, basically you are right. The option A is the answer. Cell plate is the structure which helps in cytokinesis, promoting cytokinesis in plant cells so that new daughter cells are formed. So if you look into the fold formation of the cell plate, this is how it happens. So once the telophase stage has started, right? So what happens is that there are several Golgi vesicles that are present within the plant cell. So it's basically a membrane-bound organelle, right? So as being a eukaryote, Golgi are present there. So what happens is that it accumulates at the metaphase stage, at the late metaphase stage in this case, and you can see that there is a non-uniform distribution of Golgi mass there. So afterwards, during the telophase has started, what happens is that this Golgi molecules along with the different microtubules that are present at this end actually combines together to form a particular content structure called as a what you call as a cell plate. This is how a cell plate is formed. That is, it is formed from the Golgi vesicles and microtubule assembly there. So they condense together to form this particular structural component called as Golgi vesicle there. And this particular component is called as the cell plate there. Instead of cleavage furrow that is present within the animal cell, in plant cell plate is the one that promotes the cytokinesis stage in this case. So afterwards, when the cell plate is formed, additional maturation of this cell plate take place, in which it leads to a formation of middle lamellae. There will be a primary cell wall here at this both ends, and there will also be a middle lamellae here. I will just open up my pointer here. Okay, so this is the cell plate, right? This is the cell plate. So then it undergoes maturation. So maturation leads to formation of a middle lamellae in between that actually cements together the two cells together. And also you can see the primary cell wall formation here. It is not that much distinguished here, but upon maturation it becomes more thickened, and then afterwards what happens is that cytokinesis take place because the cells are now enclosed, right? It consists of this primary cell wall on the outside, and it consists of whatever cell wall, that is cellulosic cell wall components that are present on the outside. Only then cytokinesis take place. So in case of plant cell plate is the one that helps in the formation of cytokinesis. However, in case of animal cell, this what is it? The contractile ring followed by the cytoplasmic furrow or the cleavage furrow that is formed helps in the formation of cytokinesis after the telophase stage. So I hope this is clear. So does anyone have any doubt? These are some of the basic aspect. You might know about this previously also, but let's just refresh it out. Basically. So yeah, I think it is quite clear, right? Okay, one. Okay, I think I can move on to the next question, right? Okay. So coming on to the next question. Which of the following best describes canonical centriole formation? Centriole arises spontaneously in the cytoplasm. New centrioles form next to pre-existing centriole. Centrioles are inherited from the nucleus. Centrioles only form in prokaryotic cell. Which might be the answer? Okay, I am getting option number 2. Any other option? Okay, I think all of you are agreeing with option number 2, right? Yeah, basically you are right. In case of canonical centriole formation, new centrioles arise from next to pre-existing centrioles. So basically, if you look into this past statement, centrioles arise spontaneously in cytoplasm. That is also a right answer, but this is not something related to canonical centriole formation. It is related to what is called as a de novo centriole assembly. So that's totally a different aspect. We will discuss that in the next question. So basically, it is not related to canonical centriole formation. So in this, the statement is right, but it is not related to this particular formation. So new centrioles arise from new centrioles form next to pre-existing centriole, that is basically right. They can have the ability to duplicate and all, right. And one will go into one polar end and other to other polar end. So one of the dot cell that is found will consist of that one centriole and centrosome, and other consist of the other centrosome there. So centriole duplication actually leads to a formation of new centriole there. So that's a different thing. Centrioles are inherited from the nucleus. No, it is not inherited from the nucleus. Centrioles only form in prokaryotic cell. No, centrioles are actually found in animal cells, right? It is a part of centrosomes there. So in case of ciliary movement and everything, like in case of animal cells and all, these actually play a part in there, and hence it is found in eukaryotic cell, not in prokaryotic cell. So yeah, so in case of canonical centriole formation, new centrioles form next to pre-existing centriole. So anyone has any doubt regarding this? Okay, so it's quite clear, right? So I will just move on to the next question. Okay, coming on to the next question. Which of the following statement about centriole formation is correct? Centrioles can be inherited, only be inherited from pre-existing centriole. Centriole formation is independent of centrosome. Centrioles duplicate once per cell cycle. Centrioles are randomly formed in the cytoplasm. Which might be the answer? This is related to something that we have previous question. Okay, I am getting option number C. Any other option? Okay, again option number C. So I think each and every one of you is agreeing with option number C, right? So basically you are right, centrioles duplicate once per cell cycle. It's like similar to what a DNA replication and a DNA chromatic separation means. So it only happens once per cell cycle, not more than that. Centriole duplication also take place like that. So each centriole has a tendency to like what you say, separate from like each daughter or each mother centriole there, right? So what you call that, so leading to formation of a daughter centriole there. The centrioles can be only be inherited from pre-existing centriole. That is not right, because there is also a experiment. There is also another phenomena called as de novo centriole assembly there, or de novo formation of centrioles there, that we will be discussing. And centriole formation is independent of chromosomes. Basically, that is centriole formation is independent of centrosomes. That is incorrect, because centriole is actually a part of centrosome. Centrioles, with two centrioles with the interconnecting linkers, along with the pericentriolar matrix and the microtubules that are attached to the PCM or the pericentriolar matrix, forms what is called as a centrosomes there, right? So centriolar formation is dependent on centrosome. It is not independent of centrosomes. That is incorrect. As centrioles are randomly formed in cytoplasm. So basically that was not right. Basically centriole duplication takes place once in a cycle, and it should be formed near the pre-existing centrioles if the centrosome is actually present there, and it follows the canonical form of centriole duplication. So if you look into de novo centriole formation, we have discussed this earlier, right? So we will be discussing it. So it actually refers to the process by which centrioles are assembled in the absence of pre-existing centrioles. So suppose that there are centrioles that are already present within the cell, the centrosomes are present there. So they can duplicate to form a daughter centrosome, while the mother centrosome remains intact, and one will go into one daughter cell and other will go into the other daughter cell. And that cycle continues through each cell cycle where it is duplicated once. But in case of de novo centriole formation, what happens is that when a centriole is absent, there is also a tendency for the cell to make its own centriole or the centrosome from itself without the pre-existing centriole there. However, it takes place only in the presence of certain chemical treatments or genetic modification. Suppose that there is a protein called as polo-like kinase 4 or PLK4, and it is supposed to be a key regulator of centriole biogenesis there. So if you overexpress this particular polo-like kinase 4, meaning that if you are increasing the amount of expression of this PLK4 protein, what happens is that even in the absence of a centriole, if even the centriole is removed, it can, or even if the centriole is formed, it can lead to production of more centrioles there whenever this particular PLK4 protein is overexpressed there, leading to the assembly of structurally and functionally normal centriole, which in turn lead to formation of centrosomes there. So this is because of the fact that that's why polo-like kinase 4 or PLK4 protein is considered as a key regulator of centriole biogenesis. While de novo centrioles can appear normal and capable of, however, one of the disadvantages of this is that they can appear normal and capable of duplication. They are structurally less reliable. The stability or degradation issues are much more in this kind of de novo made centrioles there, compared to those found by canonical duplication, which have been duplicated from the mother cell, mother centriole there, and which typically ensures the high fidelity of this particular centriole that is formed by canonical duplication, which is less as compared to that is formed in de novo centriole formation. And also, it is particularly prominent in post-mitotic cells like multiciliated epithelia. In case of cilia, the movement is needed there, centrosomes are needed there, so it can be formed by de novo assembly there, where a higher number of centrioles is required for ciliogenesis. So the genes necessary for centriole assembly are upregulated to facilitate the production of numerous centrioles because of the higher movement capacity of the cilia there, right? Because of the cilia biogenesis and you need to have more centriole there. So that's why de novo centriole formation also take place in the case of ciliogenesis also. Yeah, so I think this is one of the explanation that I could give from this particular MCQ. So I hope this is clear. So anyone has any doubt? Okay. So I think it is quite clear. So I think I can move on to the next question then. Coming on to the next question. So this is a basic question. Which of the following is the correct sequence of mitotic phases? So you can read the option and then directly attempt the answer. It's a basic question, right? Yeah, option 1, right? So in mitosis, it is started by prophase. Even in the prophase, there is early prophase, prophase, and late prophase, and each stage consists of that. Metaphase also there. There is early metaphase, late metaphase. In case of anaphase, there is anaphase A, anaphase B, and in case of telophase, there's a different. It is not that much distinguished there. Afterwards, it is followed by cytokinesis, right? So that marks the end of the cell division there. Then it enters into G1 phase, followed by S phase, another G2 phase, and then again comes back into this particular cycle itself. So answer to the question is prophase, metaphase, anaphase, and telophase. PMAT, the short form for this particular what is the step there. So you are basically right. Okay. Okay, so I think we can move on to the next question. So anyone has any doubt in this question basically? Even though it's basic, I'm just asking you. Okay, so I think it's quite clear, right? I will just move on to the next question then. Coming on to the next question. During which phase of the mitosis do the sister chromatids separate? Prophase, metaphase, anaphase, or telophase? Okay, I'm getting option number 3. Yeah, basic question. So I think all of you know the answer, right? So answer to this question is anaphase. So if you look into, these are different phases, right? So I will just give you a brief discussion. I know that you know this, but I will just give you a brief discussion on this. So this is the interphase cell. You can see that the chromosomes are decondensed there. Nuclear envelope is also there, and also you can see that nuclear envelope is assembled there. It is not disassembled, and there is one centrosome that is present there, right? So you can see that two centrioles that are present there. So in the prophase, what you can see that the centrosome gets duplicated, and also you can see the formation of microtubular assembly there, and also the DNA that is present there is getting condensed into what is called as a chromosome there. You can see the one chromatid there, two chromatids compose a chromosome there, and the nuclear envelope is also kind of getting disassembled, but it is getting initiated at that stage only. In the pro-metaphase, what happens is that the nuclear envelope is more disassembled there, and the centrosome starts to accompany its opposite pole, one at this region and the other at this region, and the centrosome starts to assemble itself into the equator. and this process is completed in case of metaphase. You can see that the chromosomes occupy this region, and this particular region is also called as a metaphase plate or an equatorial plate, and the centrosomes are occupied the polar region, and the microtubule is attached to each region there, like each chromosome there. You can see that in some cases it is attached to the ends of the chromosome there, like in this case, and also in some cases it is attached to the centrosome there. So there are different types of microtubules there, we will discuss before that. And this happens in case of metaphase. The chromosomes are actually present at the metaphase plate or the equatorial plate, the centrosomes at the opposite pole, and the microtubule assembly take place where the microtubules are attached to each centrosomes or the end of the chromatids, and also the nuclear envelope has completely disintegrated there. And afterwards what happens is that anaphase take place. APC or anaphase promoting complex becomes active in this stage. At the late metaphase stage, the separase enzyme is active from because it is not getting sequestrated by securin, and this cleaves the cousin molecule there, and the chromosomes are separate, and the chromatids become free there, and the microtubule starts to pull the chromatids towards the end, because in the minus end of the microtubule, right, as we have discussed before, the depolymerization take place, because the microtubule minus end is actually present here. Depolymerization take place, and this shortens in this case. As it shortens, the the chromatids actually move towards the opposite pole, towards the centrosome in this case. In both cases you can see that, towards the opposite poles. And afterwards, when anaphase take place, if it is in case of animal cells, you can see that there is a contractile ring formation there because of the activation of rho GTP there. And also you can see that the nuclear envelope starts to recondense back, and the chromatids also starts to arrange irregularly as what you see in prophase stage, and one of the cell, and there is a plate that is present in between there, meaning that the cell wall formation, plasma membrane formation is taking place there in case of animal cell, and the one cell receives one centrosome and the other cell receives the other centrosome, and then it takes place, then it divides into two, one dot cell you can see, and this is what you this enters into what you call as a G1 phase, then into S phase G2, and then again repeating the cycle of cell cycle division there. So basically I was just giving a brief explanation on this, so I hope it's clear. So does anyone have any doubt in this case? Okay, I hope it's clear. So I'll just move on to the next question then. Coming on to the next question. What would happen if the spindle assembly checkpoint or SAC fails? Cells would remain in the G0 phase. Chromosomes might not be equally distributed. DNA replication would not occur. The cells would skip anaphase. Which might be the answer? Spindle assembly checkpoint fails, or whether it is inactive in nature. Some mutation might have happened, so it fails to check whether it is happened. So okay, I am getting option number 2. I am also getting option number 1. Okay, so I will just give you the answer. So the answer to this question is option number 2. Chromosomes might not be equally distributed. So coming to option number 1, if you look into G0 phase, because this spindle assembly checkpoint has nothing to do with G0 phase. G0 phase is also called as quiescent phase or a resting phase. So in some cases, when a cell enters into G1 phase, it can enter into another cell cycle, or what it can do is that it can enter into a phase called as a G0 or a resting phase, where the cells undergo can do some metamorphosis in itself, and it can become of specialized cells that can carry out specialized functions. Suppose that there is a blood cell, a hematopoietic stem cell there that is in continuous division, right? However, in some case in G1 phase it enters into a G0 phase where it gets specialized to perform one of the function of a blood cell, like it can become RBC, it can become WBC, similarly to that. So spindle assembly checkpoint is not associated with that particular phase. That's the incorrect answer. Chromosome might not be equally distributed. So basically that's an answer, because spindle assembly checkpoint is a checkpoint where it checks the cell where the microtubules are attached towards the kinetochore that are present in the chromosomes there, right? So only when the kinetochore, it is actually attached to the, when the cell itself dictates that it is attached to the correct kinetochore, the metaphase to anaphase transition take place. So and also this also enables to leading to a formation of what is into mitotic exit network. So spindle assembly checkpoint actually checks whether the microtubule is attached to the kinetochore. Only then, once it passes that particular step, the chromatids get separated and the anaphase telophase step can proceed with that. So if it is not happening, if the spindle assembly checkpoint fails, anyway, if the microtubules are not properly attached to the chromosomes, what happens is that chromosome might not be equally distributed, because this is what the function is. It needs to promote that the chromosomes are equally distributed after cell division. If it is fails, then chromosomes might not be equally distributed. DNA replication would not occur. That's a different thing. That's not associated with this step, and cells would skip anaphase. So it's actually present before that basically in this case. So it wouldn't skip anaphase. Anaphase would happen, but what happens is that chromosome might not be equally distributed. So it does not ensures that this particular checkpoint ensures that the cell cycle does not proceed to anaphase until the chromosomes are properly attached to spindle. That's what we have discussed now. Monitors the interaction between kinetochores and the spindle microtubule, ensuring proper attachment before allowing chromosome separation. And it is active during pro-metaphase stage and is downregulated once all the cystochromatids are correctly aligned on the mitotic spindle. So once that is promoted, there is no need for this checkpoint to be active. So so all the components that promotes this checkpoint is actually getting degraded out, promoting anaphase, like what is this particular transition to anaphase stage. And it leads to a formation of mitotic checkpoint complex, and also this complex inhibits the anaphase promoting complex, which is necessary for the separation of chromatic complex. It actually activates, it actually activates. That's why anaphase is taking place, right? This is a mistake there. I will clear this out. So it activates the anaphase promoting complex, which is needed for the separation of cystochromatids there. So I hope this answer is clear. So anyone has any doubt? Okay, so yeah, okay, thank you. So I will think it is quite understandable. If you have any doubt, you can also ask later, no issue. So I will just move on to the next question. Coming on to the next question. In which stage of the cell cycle do spindle poles move apart from each other maximally? Metaphase, anaphase A, anaphase B, and telophase. Which might be the answer? In which stage of the cell cycle do the spindle poles move apart from each other maximally? Okay, I am getting option number 2. Metaphase, anaphase A, anaphase B, or telophase. I have got option number 2. Any other option? Okay. So answer to this question is option number 3, anaphase B. So basically there are two types of anaphase, two stages of anaphase, anaphase A and anaphase B. So basically like what happens in anaphase A is that chromosomes are pulled poleward. Like in case of anaphase, what happening is that it's kind of the initiation stage that is taking place there. So it leads to formation shortening of kinetochores, microtubule, and the force generated at the kinetochore by the microtubules helps in the movement of daughter chromosomes to a seed spore. You can see that there. However, however, the force that is exerted in this particular stage of anaphase, that is anaphase A, is currently less or is more reduced as compared to that of anaphase B. So from the next stage, you can see that in case of anaphase B, you can see that the poles are already moved apart there. Another level of sliding force is generated between the polar microtubules from the opposite pole to push them apart. So an excess of force is generated from this case. So the chromatids are already separated there. However, an excess amount of energy or the force is generated by the microtubule or the polar microtubules that are present there, that are present near the that are present near the poles there, which pulls the microtubules more separated from each other. and then you can see that the distance is actually getting increased as compared to that of the anaphase B, anaphase A stage that is taking place there, because of the extra energy that is exerted by the polar microtubules that are present there. That's why the in case of anaphase B stage, the spindle poles are moving apart much more maximally as compared to what is seen in case of the anaphase stage in case of anaphase B. So that's the difference between both the cases. In some cases, in this case, you can see that polar microtubules are not acting. In anaphase B, polar microtubules are acting along the astral microtubules or whatever it is, which helps in generating an excess force, which pulls the chromatids more at a more distance away from each other. So yeah, so in which stage of the cell cycle, anaphase B, in case of anaphase B, the spindle poles move apart from each other maximally. So I hope this answer is clear. So anyone has any doubt? So I think it's clear. So I will just move on to the next question. Okay, coming on to the next question. The microtubules which radiate out from the pole and helps in positioning of the spindle pole in the cell are termed as interpolar microtubule, kinetochore microtubule, astral microtubule, or all of the above, which might be the answer. It helps in positioning the spindle pole. You just need that term basically. Spindle pole is actually something that is present in the chromosomes, right, in the centrosome at that region. Okay, I am getting option number 3. Any other option? Okay, I am getting option number 2 also. Option number 3 I am getting. So actually it's option number 3, not 2. Astral microtubules, right? Spindle pole, from the term you can see, you can get the answer, right? So basically if you see, there are 3 different types of microtubule that are labeled here. One is the interpolar microtubule, kinetochore microtubule, and astral microtubule. So basically in case of this, what is a kinetochore microtubule, from the name itself you can feel that it is something related to kinetochore. So these are microtubules that radiate from the centrosome. These are the negatively charged end of the microtubule there. The positive end of the microtubule actually binds with the, what you say, the kinetochore. So kinetochore is a protein that is, there are 2 different kinetochore, inner as well as outer kinetochore. So basically these are protein complexes there. Several protein complexes make up a kinetochore there. So this what is a microtubule, this kinetochore microtubule actually attaches to the kinetochore that is attached to a centrosome. So it is directly attached to a centrosome, indirectly attached to a centrosome that is present within this chromosomes there. While in case of interpolar microsome, you can see that there are several motor proteins that are present here. Majority of this, primarily it would be actin motor proteins, sorry, kinesin motor proteins there, because it is occupying majority of this region there. So it is not directly attached to the, it is present at the the equatorial position, similar to that of the kinetochore microtubule. However, it is not attached to the kinetochore. However, it is attached to these motor proteins that are present in between, not to the chromosomes there. And also that's what are called as interpolar microtubule, because it is present at this polar region. However, it is not getting attached to the chromosomes there. And the other one is the astral microtubule that radiates out from this region to the negative end of this particular cell there. So they are present at the spindle pole rather than the equatorial region there, spindle equator there. So that's the answer to this question. The microtubule which radiate out from the pole and helps in positioning of spindle pole in the cells are termed as, what is a astral microtubule. So I hope this answer is clear. So anyone has any doubt? Okay. Okay, so I think it's quite clear, right? So I will move on to the next question then. Okay, so how many microtubule does the budding yeast kinetochore binds to? 1, 2, 3 or 4? This might be the answer. How many microtubule does the budding yeast kinetochore bind to? I'm getting option number 2. Any other option? Okay, so answer to this question is 1. So a kinetochore, budding yeast kinetochore only binds to, yeah, you are right, basically one of first option. Budding yeast kinetochore only binds to one microtubule. So basically this is the whole interaction that is taking place there. So you can understand this chromosome, a portion of the chromosome, the centrosome portion of the chromosome there. Sorry, the what is it? The central portion of the chromosome there. And this is the inner kinetochore. So there is one outer kinetochore also, inner kinetochore also. So microtubules have a tendency to bind to the inner kinetochore there. And you can see that the inner kinetochore is also made up of several different kinds of protein there. So it's not just a single protein, it is actually a complex of different proteins there. You can see that this is the microtubule and this is the DAM1 complex. Actually DAM1 complex is a complex that actually helps in the anchoring this particular microtubule to this kinetochore there. And along with the help of other protein complex called as NTC80 complex, SPC105 complex or MIC12 complex. So you can see that the NDC20 actually acts as a long flexible anchor that helps in the interaction of DAM1 complex with the SPC105 complex or MIS12 complex. And this MIS12 complex has the direct interaction with the inner kinetochore complex. So I think there's not that you have to go much detailed into this particular process that is taking place. However, you should know what the interaction or kind of complex that are present there and how they are basically interacting there basically. You see the microtubules are attached because of this particular whole protein complexes to the kinetochore there. And yeah, so yeah, that's how the spindle assembly actually take place there. So only one microtubule does bind to a budding yeast kinetochore. So can any one of you tell me what is a budding yeast? What is the scientific name or scientific nomenclature of the budding yeast is? What is the scientific nomenclature or the biological nomenclature of a budding yeast or a fission yeast? You can just type it out in the chat box, no issue. Yeah, Saccharomyces cerevisiae, right? So yeah, it's a spelling mistake, basically no issue. So basically, budding yeast is also called as a Saccharomyces cerevisiae, while the fission yeast is also called as a Schizosaccharomyces pombe, right? So in case of fission yeast, the bud formation does not take place. Similar to what happening in a plant cell, there is a plate formation there or a cleavage furrow that is formed there, and then the cell, elongated cell, actually divides into two daughter cells. In case of Saccharomyces cerevisiae, which is obviously called as a budding yeast, bud formation take place. From that bud, actually two daughter cells are formed there, right? So the bud enlarges within each stages of cell cycle with each, when it is passing through each cell phase, and then it leads to a separates out and leading to the formation of two daughter cells. That's why one is called as budding yeast and the other is called as fission yeast. So I hope this is clear. So anyone has any doubt? Okay, so I think it is clear, right? So I will just move on to the next question then. Okay, coming on to the next question. The transition from shrinkage to growth phase for microtubule exhibiting dynamic instability is termed as assembly, catastrophe, disassembly or rescue. Which might be the answer? Transition from shrinkage to growth phase of a microtubule. Microtubule actually pass through different phases, right? Which might be the answer? Okay, so answer to this question is rescue. So basically this is how a microtubule functions, right? Microtubules have a tendency to assemble itself and it also have a tendency to disassemble itself, right? So if it is undergoing assembly, meaning that it's undergoing a phase of growth where this particular end of the microtubule is getting transferred by either a GTP or an ATP. Because of the ATP or GTP hydrolysis taking place there, polymerization happens. So this actually undergoes a process called as growth. This particular assembly stage is called as a growth. However, it can enter into a stage of catastrophe where the disassembly of microtubule actually take place there. So the initiation stage of the disassembly of microtubule is actually called by a phenomenon called as catastrophe. So when the catastrophe actually continues out, the whole growth, the whole pattern that is taking place is determined by a phase called as a shrinkage there. You can see that from the growth it enters into a stage called as catastrophe. So during the catastrophe it shrinks in size. No, it doesn't grow, but it shrinks in size, and therefore it is called as a shrinkage phase or a disassembly phase there. So when a disassembly phase reaches a particular limit, or when the microtubule or microfilament assembly microtubule assembly needs to take place, what happens is that it undergoes a phase called as rescue, meaning that again the GTP is actually getting, these tubulin dimers are actually bound by GTP there, and it comes again into this particular phase, and then it initiates a phase called as rescue, where the microtubule actually enters from a shrinkage phase to a growth phase. It initiates that particular phase, and that particular phase is called as a rescue phase there. So the transition from shrinkage to growth of the microtubule actually exhibit a stage called as a rescue phase, not a catastrophe phase. Catastrophe is the totally the opposite phase, that if it transitions from a growth phase to a shrinkage phase, then it becomes a catastrophe. If it transitions from a shrinkage phase to a growth phase, it's called as a rescue phase, where the polymerization of microtubule reinitiates again. So that's why it's called as a rescue phase. And it reinitiates, not initiated. If it is initiated, it's called a growth phase. If it is reinitiating back, it's called as a rescue phase. So I hope this answer is clear. So anyone has any doubt? Okay, so I think it's clear, right? So I will just move on to the next question. So coming on to the next question, which of the following scenarios would lead to the active mitosis exit, that is active CDC50, would not lead to active mitosity, active CDC50, active DBS2, MOP2, MOP1 complex, sequestration of CDC14 by NET1, inactive BUP2, BFA1 complex? Which might be the answer? Which of the following scenarios would not lead to an active mitosis exit? Okay, so I will just give you the answer. So answer to the question is option number 3, sequestration of CDC14 by NET1 complex. So basically this is the whole process. This is one step and this is the other step. Two different diagrams are there, but it leads to formation of like a same mitosis exit there. So nomenclature of certain proteins differ in this case. That's why I thought to show two diagrams there. So what happens is that if you look into this particular diagram, right? So basically what happens is that CDC14 is actually bound to a protein inhibitor called as a CFI1, or in case of this you can see that CDC14 inhibitor is called as NET1. So in case of organism that nomenclature will differ. In case of humans it would be NET1, in case of Saccharomyces it will be CFI1, right? So that will be different. So afterwards what happens is that when mitosis needs to be started, so like in the metaphase stage it's actually initiated, this CDC5 levels get in like increased in that case. So CDC5 is actually like a kinase there. So what it does is that it phosphorylates one of the region of CFI1 or NET1 in this case. So what happens is that when it is getting phosphorylated, it is recognized by this particular CDK, that is cyclin B as well as CDK. So cyclin B and CDK are present in the metaphase stage, right? It is actually it is also called as a mitotic cyclin. So it recognizes this phosphorylated amino acid that is present in CFI1. So when it is bound to this CFI, this particular CFI1 inhibitor complex, what it does is that the kinase, the CDK kinase that is present within the cyclin CDK complex actually phosphorylate another amino acid within the CFI1, thereby thereby changing the structure of CFI1. So when it is happening, the CDC14 that is bound to this particular inhibitor complex is getting released there. So it is now free there. It is not active yet. It is however free. So what it can do is that another protein complex, either it can move into the cytoplasm there, into the spindle body complex that are present there. It can move out of the nucleus to the cytoplasm there. Or what it can do is that if it is another complex, when these two complex amino acids are phosphorylated there, right, DBF2 or MOB1 complex that is present within the spindle polar body. Spindle polar body is actually found consists of centrosome and everything in that case. So this is getting activated there. This moves from this spindle polar body to cytoplasm to the nucleus, and this recognizes this CFI1 complex. Once it is recognized, it means that it indicates that the CDC14 is active. So this complex can either phosphorylate it directly within the nucleus, or the CDC14 can move from the free CDC14 can move from the nucleus into the spindle polar body, and then it can interact within the DBF2 or MOB1 complex, or it can get phosphorylated outside. So, so in both cases, what it means that the CDC14 that is present within the nucleus, which is getting phosphorylated, coins the cell to exit from mitosis, or which is present within the cytoplasm, which is phosphorylated, can interact with the other complexes that are present that are needed for cytokinesis. So both leads to this particular mitosis exit. So that's why it's called as a MEN network, that is mitosis exit network. And also this whole process starts with this particular complex called as I think it is this CDC14, CDC15. In alphabetical order it started with an F, right, the 15. CDC15 early anaphase release complex. So this whole process starts with this, then it ends with the mitosis exit network there. So you can see the other diagram also, it is similar in case, but some more elaborate coins are termed there. In some cases the SPO12 protein complex are activated there, which inhibit this FOB1. If FOB1 is active, it means that it cannot release the CDC14 from net1, or it cannot the CDC14 is inactive there because bound to net1. So similarly CDC5 is also activated there. You can see that CDC5 phosphorylate this net1, and then it is recognized by the cyclin B CDK complex, thereby releasing the CDC14. Similarly CDK also there. You can see that the CDK that is present there, cyclin B CDK1 there, right, and HIT1 RSA1 protein is also there. So this is a multi-interactory regulatory pathway taking place, leading to the formation of active CDC14 there. So it can lead to other activation of other CDC15 complexes there. Active CDC15 levels are increased more in this case, and also MOB1 DBF2 complex is also active there. So these two options are right. In this case, it is not incorrect. It should be active, leading to formation of mitosis exit network, and inactive BUB to BFA complex. So you can see that it needs to be inactivated there, right? So if it is active, you can see that TEM1 is inactivated there, which is a GTP binding complex there. If it is inactive, it cannot activate CDC15 there. So it needs to be inactive there. So this particular step is also right there. So yeah, so I think this is actually a complex network. So it's actually difficult to understand at first, but if you look into a diagram over and over again, you could know how it is actually functioning. So that's why I thought to show this whole diagrammatic step there, because you could gain more knowledge into what's happening there. So I think this you actually know the answer to that. It doesn't need to sequester net1, because net1 is the inhibitor of CDC14. If it is sequestered to net1, it won't allow mitosis exit. It needs to separate itself from the net1 in order to become active, leading to active mitosis exit. So I hope this answer is clear. So anyone has any doubt? And because the CDC 14 is getting activated there, right, it can activate this CDC 15, which in turn goes to the spindle polar body, activating DABF2 MOB1. That's why when the CDC 14 is coming out, it is getting phosphorylated there, leading to cytokinesis. So that's whole interconnected step there. So I think this is clear. So I think I can move on to the next question then. Coming on to the next question. Okay. So exit from mitosis is followed by nuclear envelope reassembly and chromosome decondensation, nuclear envelope disassembly and chromosome decondensation, nuclear envelope reassembly and chromosome decondensation, or sister chromatid dissociation, which may be the answer. Okay, I'm getting option number 1. Any other option? Okay. So answer to this question is you are right. Basically, it leads to chromosome envelope reassembly and chromosome decondensation, right? So basically what happens is that in case of before the mitosis started, like chromosome nuclear envelope disassembles and chromosome starts to condense, right? So whenever the mitosis has happened or the cytokinesis has happened, the at the telophase stage what happens is that the nuclear envelope that has been disassembled when during the start of the prophase stage start to reorganize itself, it starts to reassemble itself, and also the chromosome that has been condensed starts to decondense itself into form DNA strands and all. So that actually take place in once the cells start to exit from mitosis, and so the answer to this question is at the telophase stage nuclear envelope reassembly and chromosome decondensation take place. So I think the answer to this question is clear. So anyone has any doubt? Okay, so I suppose it's clear. I hope it's clear. So I will just move on to the next question. Okay, centrosome replicates once per cell cycle, twice per cell cycle, 4 times per cell cycle, or never. Which might be the answer? We have discussed this before. Yeah, once per cell cycle I am getting. Yeah, basically you are right. So one of you is right. So it actually replicates once per cell cycle. Okay, coming on to the next question. The microtubule depolymerizing drug nocodazole can arrest the cell in G1 phase, S phase, G2 or M phase, G0 phase. Which may be the answer? Nocodazole. Which phase will be it affect? It's actually a depolymerizing drug, microtubule depolymerizing drug. Okay, so it actually arrests G2 to M phase transition. It doesn't affect the phase, but what you say, because of this activity, it promotes the transition from the G2 phase to M phase, mitotic phase. So basically, it is actually called as an anti-neoplastic agent there. So do check what is called as an anti-neoplastic agent there. Why it is called as a neoplastic agent there? You get the answer. And primarily known for its ability to disrupt microtubule disassembly, because it is a microtubule depolymerizing drug, right? So it affects the destabilizes the microtubule dynamics. Therefore, release of tubulin molecules take place, and then it actually helps in depolymerization there, and including cell division, which are crucial for various cell processes, including cell division. So what it does is that it binds to beta tubulin that are present within the microtubule there. So there are different types of tubulins that are forming a microtubule. Alpha is there, beta is there, delta is there, and one more is there. There is also a phosphorylated gamma tubulin there, and also epsilon tubulin is also there in some cases. So it actually binds to this beta tubulin, inhibiting the polymerization of microtubule. So because this beta tubulin and alpha gamma tubulin are actually needed to promote polymerization. So if it binds to that, then it cannot bind to a microtubule, and it promotes depolymerization of microtubule. So this binding prevents the assembly of microtubule from tubulin dimers, leading to the disassembly of preformed microtubules in the cell. So that's the basic mechanism of this particular drug. And by disrupting microtubule formation, nocodazole prevents the proper formation of the mitotic spindle during the cell division, because spindle formation is required so that microtubule can attach to a spindle body and also to a centrosome. Later, depolymerization of microtubule happen place once this spindle is actually attached to kinetochore, thereby pulling the chromatids away from the equatorial plate to the polar plane of the cell there, right? So that's why metaphase to anaphase transition actually take place. So if this microtubule is not formed there, the cells treated with nocodazole can enter mitosis but fail to complete its due to inability to form functional tubules, resulting in this particular phase arrest, G2 plus M phase arrest, because it can enter into a early mitosis phase, but completely it cannot enter into a mitosis phase. So it is actually arrested between this particular phase there. The G2 to M checkpoint actually detects this particular malfunctioning because of this particular drug there, thereby preventing the phase arrest, thereby enabling the arrest there, preventing the complete transition from G2 to M phase there. So the absence of microtubule attachment to kinetochore activates the spindle assembly checkpoint, causing the cells to halt the progression in the prometaphase. So that's what happens there. Basically, the spindle assembly checkpoint detects that the microtubule is not getting assembled there because of the presence of this drug. So it actually activates this checkpoint, and hence it is getting stuck between the stage, that is the G2 to M phase, prometaphase stage there. And it can also trigger apoptotic pathways after inducing mitotic arrest, because the spindle assembly checkpoint is getting activated there, right? So the cell is considered faulty there. So other factors like P53 is activated there, and this PARP complex is also getting cleavage there. Caspase are activated there, which are apoptotic proteins, right? There are caspase, procaspases, anti-apoptotic caspases there, which actually leads to the formation, reduction of this anti-apoptosis proteins, leads to this apoptosis pathway. So the cells get degraded out afterwards because of this malfunctioning pathway, that is arrest that is taking place there, that is the G2 to M phase. So nocodazole is an anti-neoplastic agent that binds to this beta tubulin, thereby inhibiting the polymerization of microtubule. So when this happens, it gets arrested at this G2 to M phase, because of the mitotic spindle checkpoints that is getting activated there. So when it is activated, it triggers the cell to undergo apoptosis because of the elevation of P53 and other caspases that is taking place there, leading to cell death. So that's the how mechanism of this nocodazole take place. So coming to azole, basically you can see that the azoles are actually antifungal drugs, right? Ketoconazole are there, fluconazole, miconazole are there. You would know that basically. So I'm not sure whether this is used to treat any kind of fungal diseases, but from the name itself, azole is there. So do check it out if for your knowledge. Because basically I saw this right now, so I thought that might be azole drug. Yeah, it could be. So I think this answer is clear. So anyone has any read out to this answer, or should I repeat it once more? Okay, so I think it's quite clear, right? So I will just move on to the next question. Okay, coming on to the next question. Which of the following cell cycle checkpoints ensure that cell do not enter anaphase until all the chromosomes are properly biooriented at the attachment to opposite M-meiosis? or a microtubule organizing complex. So intraphase checkpoint, DNA damage checkpoint, spindle assembly checkpoint, spindle position checkpoint, which may be answer. Okay, so the answer to this question is spindle. Yeah, option number 3. Yeah, you are right. Basically, option number 3 is the right answer. Spindle assembly checkpoint. So yeah, other checkpoint, DNA damage checkpoint is there. Intra-phase checkpoint I also don't know. Better check it out. And I think option number 4 is just a different name, just to confuse out and all. Spindle assembly checkpoint we know. So answer to the question is spindle assembly checkpoint. Okay. So coming on to the next question. Which of the following is a target for APC during anaphase? Securing, CDK1, CLB5 or separase? Which may be the answer? Which is a target for anaphase promoting complex during anaphase? Okay, I am getting option number 1. Option number 2 I am also getting. Okay, so answer to this question is option number 1, securing. Basically, CDKs are not targeted by APCs. So what APCs target is are cyclins. CDKs are actually present like at a particular limit at all points of the cycle. But however, cyclins, when it bound to a cyclin, it becomes active, right? It has a kinase activity, but it is inactive. Only after binding to a cyclin, it becomes active. So securin is actually a component that is bound to separase, right? So when it is bound to separate, it acts as an inhibitor. So whenever anaphase need to start, an APC or anaphase promoting complex binds to phosphorylate this securin, it enters into what is called as the ubiquitin degradation pathway. So it is tagged by polyubiquitin complex there, which is recognized by this proteasome, which then later degrades out. So when that happens, separase remains free. Separase binds to cohesin, degrades it out. It separates the chromatid so that the chromatids can move to opposite poles, thereby initiating the metaphase to anaphase transition there. So that's what happens there. And I also expect that you will look into this particular option also, CLB5, sorry. It's actually recognized as cyclin B5. So cyclin B5 is a type of cyclin B. It is also involved in metaphase to anaphase transition, and from what I read, it is also targeted by this anaphase promoting complex. So cyclin B needs to be degraded out, right? Cyclin B needs to be degraded out from CDK1 by polyubiquitination and followed by proteasomal degradation. So it is also recognized by this APC. So whether try to find out whether this securin along with the CLB5 is also an answer. So from my point of view, securin is a probable answer, but CLB5 can also be an answer. So do check it out also. So I hope this is clear. So anyone has any doubt? Okay, so I think this answer is quite clear. I'll move on to the next question then. Do check it out if CLB5 is also an answer. Okay. So, which kinase help to attach sister chromatids to the spindle? Polo-like kinase, Aurora kinase, Aurora B, Aurora kinase, Aurora A, or none of the above? Which might be the answer? Anyone know the answer? Okay, I think I will just show you the answer. So the answer to this question is Aurora kinase, Aurora B. So polo-like kinase, as we have discussed before, one of the function of polo-like kinase is that it can lead to de novo generation of centrioles. If centrioles are absent there, if polo-like kinase are getting overexpressed there, it can lead to de novo assembly of centrioles there, formation of new centriole, even if pre-existing centrioles are absent there. In case of coelogenesis, we have discussed that before, right? It is actually important. So in this case, it is important for also important for the assembly of bipolar mitotic spindle. It phosphorylate proteins required for separation of spindle poles early in mitosis. So it doesn't help in the attachment of sister chromatids. However, it phosphorylate proteins required for the it helps in the separation of spindle poles earlier in the mitosis. And Aurora kinase Aurora A is essential for assembly and stability of the spindle. So spindle, it helps in the assembly and stability of the spindle, like the microtubule assembly and everything, and it also helps in maintaining stability of microtubules there. However, it doesn't help in attachment of sister chromatic to a spindle. So it is the function of the Aurora kinase Aurora B that helps in the this particular function, that is Aurora B actually helps in the this kinase actually helps in the attachment of sister chromatic to a spindle. So I hope this answer is clear. Anyone has any doubt? Actually, polo-like kinase is actually from the name itself is a kinase, right? It can phosphorylate other proteins. So it's actually a part. It's also a part of this mitotic spindle. It helps in the separation of spindle poles early in the mitosis. So basically, like when the decondensation of chromosomes happens, right? So earlier phases, there is a need that the spindle folds need to be separated out. So it needs to know that this is the pole where the microtubule need to be generated out. So this polo-like kinase has a function to carry out this particular character of this protein. And also from the previous step, and also other than that, I can show you one other. We have discussed this earlier, so but I will just show you that. So there is also, like, you can see the polo-like kinase. Another function of polo-like kinase is there. One is the distinguishing the spindle poles there in the early mitosis phase, and also it also leads to formation of centrioles there. So in case of canonical centriole formation, what happens is that from the pre-existing centriole that is present there, new centrioles can form. One daughter centriole can form, leading to formation of two centrosomes there. In case of de novo centriole, what happens is that if the pre-existing centrioles are absent in the cell, the centrioles can also form. So what such example is that overexpression of polo-like kinase 4, which is a key regulator in centriole biogenesis, can lead to the formation of de novo formation of the centriole, even if the pre-existing centrioles are not formed there. Especially in this case of coelogenesis, in case of cilia formation and all, right? Centrioles are needed because centrioles actually helps in microtubule assembly and disassembly, right? Cilia actually needs this microtubule assembly and disassembly there, and thereby de novo centriole formation actually take place in this kind of region where multiciliated epithelia needs to form there, leading to the formation of higher number of centrioles there, which is essentially in coelogenesis. So one such example of function of polo-like kinase is the formation of de novo centriole, and also enabling the recognition of mitosis spindle there, that it helps to distinguish yeah, it helps to distinguish the separation of spindle poles early in the mitosis. So I hope it's clear. Kinase, it's actually phosphorylate proteins. So is the doubt clear, or should I explain it once more? Okay, so I think I will just move on to the next question if anyone has any other doubts. If anyone of you don't have any other doubts, okay. Okay, coming on to the next question. Which type of microtubular assembly is seen in centrioles? 9 duplet of microtubule, 9 triplet of microtubule, 10 duplet of microtubule, or 10 triplet of microtubule? Which may be the answer? Yeah, option number 2 I am getting. Any other option? Yeah, I think every one of you go with this option, right? 9 triplets of microtubule. So this is the structure. You can see that this is a centriole there, and you can see that this particular structure, this is the enlarged structure there, right? So this is a microtubule triplet. There are three different microtubule that comprise there. That's why it's called as a microtubule triplet. So in more scientific term, it is this structure is called as a protofilament. This whole structure is called as a protofilament. So it is actually a triplet of microtubule there. So if you count the how many triplets are present there, you can count that 1, 2, 3, 4, 5, 6, 7, 8, 9. There are 9 triplets of microtubules there that comprises a centriole there. Apart from that, other components like a central hub is present there. A spoke region that connects the central hub to the microtubule is present there, and each protofilament is connected to each other by an ACC linker there. So yeah, so that's the whole structure of a centriole there. Yeah, 9 triplet of microtubule. There are 3 microtubules there comprising protofilament, and there are about 9 protofilaments. So hence there are 9 triplets of a microtubule that contributes a centriole there. So does anyone have any doubt? This is a common structure seen in centrosome. So I hope it's clear. So I think that I can move on to the next question. Okay. This we have discussed before. Surprise cleaves which part of the coercion complex? SMC1, SMC3, SCC3 or SCC1? Which might be the answer? Coercion is actually linking two chromatids together, right? So there should be something that needs to be cleaved out from the coercion molecule so that the coercion will be degraded out and the chromatids will be free, so that it can transition from metaphase to anaphase stage and the chromatids will move to opposite poles. Which part of the coercion complex does surprise do? Surprise cleaves the surprise promote that because it's activated by APC. However, it cleaves only a certain part of this coercion molecule. Which might be that be? Sorry, coercion complex. Okay, I can give the answer. So SCC1 is the part of the coercion molecule that is actually getting degraded out from the degraded out by the separate molecule. So you can see that this is structure. There is a SMC3 structural maintenance of chromosome 3 protein, which is actually dimerized together, and it is combined with the SMC1. So if you just compare these two, it acts like a clipper molecule there, right? And these clipper molecules are held together by this particular SC1 SEC monomeric ACC1 complex there. So what separates this is that once it is like separate from or away from the securing molecule, which is degraded by APC complex by proteasome degradation mechanism, this separase recognizes this SC1 complex, it cleaves it out. So now you can see that like this complex is free. It only consists of this SMC3 as well as SMC1 complex, and the clipper form of the or the separate coercion molecule is actually open there. So the when it is open, it is free and it releases itself from the region where the it has earlier bound to the chromosome, keeping the two chromatids intact to form what is called as a chromosome there. And then the chromatids are actually free. So the microtubule disassembly take place. Then it because of the action of the like polar microtubule and other motor proteins there, the chromatids are getting separated from each other because the coercion molecules are in open form and it is not at all intact now. So the separase molecule actually cleaves this SCC1 complex of the coercion molecule. Coercion molecule there. Sorry, this is a mistake there. So others are component of the coercion molecule, but it is not acted upon by this separase enzyme complex there. So anyone has any doubt regarding this? Okay, so I think it's quite clear. So I just move into the next question then. Coming on to the next question. Which part of the centrosome microtubule assembly. It nucleates microtubule so that assembly can take place. Alpha TURC, beta TURC, gamma TURC, or all of the above. Which might be the answer? Which of these particular tubulins that are present in the centrosome actually nucleates, or the centriole actually nucleates, leading to microtubule assembly? Okay, I am getting option number C. Any other option? Okay, so basically you are right. Answer to this question is gamma TURC. So gamma tubulin is actually a component of this gamma tubulin ring complex, that is gamma TURC, which serves as a microtubule nucleator at the microtubule organizing center, that is MTOC, like the centrosome. Centrosomes actually consist of the centrioles. Centrioles consist of these tubulin networks, right? Consist of this microfilament, and microfilament consist of these tubulin networks there. So this gamma TURC facilitates the assembly of microtubule by providing a template that mimics the structure of microtubule protofilaments, enabling the addition of alpha and beta tubulin dimers to initiate polymerization. So this gamma TURC that is present at the centrioles, actually what happens is that it actually acts as its template that it sequestrates this alpha and beta tubulin dimers to its region because of the charges held together by it, and it initiates polymerization. And this activity or this nucleation process is essential for establishing the microtubule network necessary for cellular functions like mitosis and intercellular transport. That's why centrosomes are needed, right, for mitosis, and as compared to this microtubule assembly motor proteins, along with the motor proteins, they are involved in intercellular transport. Apart from that nucleation, it contributes to a spatial organization of microtubule also. How the microtubule needs to be arranged, from where to where it needs to be arranged. It also has a tendency to carry out that function, and it creates a microtubule free zone at the center of centrosome, which is important for maintaining the proper microtubule arrangement and function. You can just read that. And this organization is vital for ensuring that the majority of microtubule extends outwards from the centrosome, forming a polarized array that supports cellular process such as mitotic spindle assembly and cytokinesis. Because you can see that near the centromere, most of the negatively charged end of the microtubules are present there, while the positively charged end of the microtubules are radiating out apart from the poles, right? So in the poles, the negative charge end of the microtubules are present there, while the positive charge end of the centrosome are near to the the centrosome, and these are called as astral microtubules, right? So yeah, so that's the basic idea. It also ensures that polarity is there so that the motor proteins can move along the microtubule film assembly also. Apart from nucleation, it also have these additional functions also. So I hope this answer is clear. So anyone has any doubt? Okay, so I hope this is clear. So I will just move on to the next question. Okay, coming on to the next question. GTP hydrolysis take place in which part of the tubulin complex? Alpha tubulin, beta tubulin, gamma tubulin, or all of the above? Okay, so yeah, so I think the answer to this question is, yeah, you are right, basically second option, beta tubulin. So this is the structure, this is a PDB structure of the complex. You can see that this is the alpha tubulin, which is recognized in blue, and the gamma tubulin, which is given in red, right? So you can see that this is the ATP molecule. You can see that to both, GTP molecule, to both subunits, the GTP actually binds. In case of, if you consider alpha subunit, it is much more of a tight linkage, So in case of beta subunit, it must make kinds of like loose interaction and all to ATP. So the tightness differs in both case of alpha beta subunit. However, in case of beta subunit, GTP hydrolysis take place. In case of alpha subunit, it does not take place. Might be the fact that this alpha subunit might be acting as a sequestering component of GTP, so that it can later become transferred into beta tubulin. However, it needs to be checked out. So this ATP is getting hydrolyzed within the beta tubulin. So so once it is getting hydrolyzed, it consists of this GTP, right? So that act as a, so the tubulin consists of a GTP, then act as a positive end of the microtubule, right? So when it is hydrolyzed, when it is bound to GTP, what is that it binds to gamma tubulin, initiating the nucleation, because gamma tubulin is the nucleating factor of the microtubule or the centriole, right? So when it needs to nucleate itself, the GTP actually binds to beta subunit, and then this alpha beta subunit actually interacts with the this gamma tubulin, therefore promoting the microtubule assembly there. So that's the basic functionality of this particular complex there. So anyone has any doubt? So okay, so I think the answer is clear. So I will just move on to the next question then. Coming on to the next question. Which one of the kinesin motor protein is minus end directed? Kinesin 5, kinesin 14, kinesin 4 or kinesin 10? Which might that be? Okay, I think I will just give you the answer. So answer to this question is kinesin 14. So basically you can see that the majority of the kinesin proteins are plus end directed. However, there are essential like some of the exceptions like kinesin 14, and there was also a kinesin that we discussed before in one of the previous questions there, and these are basically minus end directed. And apart from that, all the dynein proteins, motor proteins that we know till now are minus end directed. So there are exceptions in kinesin proteins. Majority are plus end directed. However, there are also kinesin molecules or superfamily of proteins that are minus end directed. So in this case, kinesin 14 is minus end, while the other three are kinesin 5, 4 and 10 are plus end directed. So does anyone have any doubt regarding this question? Okay, I hope that is clear. I will just move on to the next question then. Coming on to the next question. Which CDC complex activates as anaphase promoting complex? CDC5, CDC2, CDC20 or CDC1? Which might be the answer? There is an activating CDC there which activates APC. When APCs are activated, cyclin B is degraded out, securin is degraded out. Yeah, these all which helps in metaphase to anaphase transition. But however, there is a cell-dependent cyclin, cell division cyclin that activates APC. Which might that be out of this option? Okay, I'm getting option number 2. So, but the answer to this question is option number 3, CDC20. Yeah, you are right basically. So this CDC20 actually activates APC, thereby regulating the other downstream pathway that are needed in metaphase to anaphase transition. So I hope this answer is clear. So I will just move on to the next question. So this is the basic the diagram there. You can see that the CDC20 is there. Basically, what happens is that when it is phosphorylated, it is inactive. When it is dephosphorylated, what happens is that it has an APC by APC-C binding protein site, which is recognized by this APC. So when it happens, this APC becomes active, and then it leads to destruction of cyclin B or securin, whatever it is necessary. So some of the CDKs and as well as MAT2 proteins actually inactivates this CDC. So when it is released from that and when it is dephosphorylated, what happens is that it binds to inactive APC, thereby because of the binding motif that is present there, thereby making it active, leading to a downstream pathway. So okay, so I think this is clear. I will just move on to the next question. So okay, which of the following have a tendency to sense spindle body for spindle polar body position? LTU1, CDC14, TEM1 or BUP2? Which might be the answer? Okay, so the answer to this question is TEM1. So TEM1 is basically a GTP exchange factor. It can hydrolyze GTP basically. So if you look into this mitosis entry network, you can see that the TEM1 is there. So this BUP2 BFA1 complex is actually inactivated there, leading to the activation of TEM1 because it is active as inhibitor of TEM1. So it can bind to GTP, hydrolyze it, and when it is bound to GTP, hydrolyze, and it can sense whether, and because of the hydrolysis that is happening here, it can sense the spindle polar body position there. So when it is active, it can lead to the activation of other complexes like CDC15, leading to mitosis exit network and all. So that is one complex that is having like this function. LTE1, you can see that it has another function there, and also CDC14 is you can see that it also has another function. It can lead to mitosis, but however, it cannot sense whether the spindle polar body position is actually there. Only when the spindle polar position body position is at the particular poles at that polar region itself, only then mitosis can exit can take place. So that is sensed by this TEM1 complex. Okay, so coming on to the next question. In Saccharomyces cerevisiae, bud formation initiates at which phase of the cell cycle? After start checkpoint, G2 or M checkpoint, after cytokinesis, or at G1 phase? Which might be the answer? Okay, so I think I will just give you the answer. So answer to the question is after start checkpoint. So start is actually checkpoint that actually happens because it's near for the transition from G1 to S phase, right? So basically what happens is that after the start checkpoint, you can see that before the start checkpoint, cytokinesis has take place and new daughter cell are formed at the G1 phase. So once it pass the start checkpoint, meaning that it can enter into another phase of DNA duplication. What it means that it starts to form a small bulb there. You can see that the bud is formed there, and in the later phase the bud actually gets elongated in the size, and also in the cell division new daughter cells are formed, and it passes through cell cycle, new daughter cells are formed, and then it again enters into G1 phase, and then it again starts into another phase and initiates the bud formation process and all. So in Saccharomyces cerevisiae, bud formation initiates after the start checkpoint when it enters into the S phase of the cell cycle. So I hope this is clear. Anyone has any doubt? Okay, so I think it is clear. So I will just move on to the next question. Which drug acts as an inhibitor to microtubule? This is the last question. Which drug acts as an inhibitor to microtubule assembly by capping the ends of filaments and enabling its depolymerization? Nocodazole, which I have discussed before, Taxol, colchicine, or none of the above? Which may be the answer? Okay, I am getting option number 4. Any other option? Okay, so the answer to this question is colchicine. So basically, colchicine caps the filament ends and depolymerizes microtubule, and because it binds to a tubulin at the polymerizing end, that is the plus end of the tubulin or microtubule, blocking the polymerization process and preventing it from formation of microtubule. Because at the plus end of the microtubule, polymerization starts. Right? If the colchicine is bound to that region, then the assembly is not for the formation of new microtubule, or the assembly is not getting activated there. So it actually inhibits the microtubule assembly there. While in case of Taxol is a microtubule stabilizing drug, it is also formulated as a commercially as paclitaxel, which binds to the taxane site of the beta tubulin. It is the site where this Taxol binds to, and it stabilizes the microtubule. It does not disassemble or destabilize the microtubule. It stabilizes the microtubule, thereby reinforcing microtubule and prevent the depolymerization of the microtubule. Nocodazole binds to a tubulin subunit that we have discussed earlier, to a gamma tubulin binds, right, and then depolymerizes the microtubule. So in this case, colchicine acts as an inhibitor to microtubule assembly by capping the ends of filament and enabling its depolymerization. Okay, so anyone has any doubt regarding this? Okay, so if there is any note and doubt, so this is the last question. So I think, do anyone has any general doubt throughout the whole topic that is covered today? So if there is not in doubt, I think we can just wind up the section for today. So thank you all for joining. So we will meet in the next class. Thank you.